Good morning, everyone. My name is Maddalena, and today I will be presenting my thesis project titled Assessing the Applicability of Novel Molecular Tools for the Taxonomic Resolution of the Mytilus species complex in Irish waters. I carried out this project at the Marine and Freshwater Research Centre at GMIT under the supervision of Luca Mirimin and Sergio Stefani. So, biological invasions are described as the colonization of a new environment by a species that was not present there before. They have been happening for centuries, and they are eased by numerous anthropogenic pathways, including ballast waters, fouling, aquaculture, and recreation or ornamental use. Biological invasions are a cause of concern because they can have ecological evolutionary consequences on the environment where they are invasive. They can also lead to genetic exchange and pollution through hybridization and introgression events, and they can also impact the local economy. The Mitigative Family is a family of saltwater edible mussels, which are benthic filter-feeding mollusks with a planktonic larval stage. In the Northeast Atlantic Ocean, the Mytilus species complex is formed by Mytilus edulis, Mytilus galloprovincialis, and Mytilus trossulus. These species are known to hybridize wherever they coexist, and a very important aspect for this thesis project is that they are morphologically indistinguishable. In particular, Mytilus galloprovincialis, which is originally from the Mediterranean Sea, is considered a non-Indigenous species in Ireland, where it has created a hybrid zone with the local Atlantic mussel. This map is from a paper published in 2020 by Lynch and colleagues, and it represents the current distribution of Mytilus edulis, here represented in black, Mytilus galloprovincialis, here represented in white, and their hybrids, here represented in grey, along the Irish coasts. So as we can see, on the Irish Sea and on the north coast, Mytilus edulis is predominant, while on the southern and eastern coasts, both Mytilus galloprovincialis and the hybrids can be found as well. Now, the presence of Mytilus galloprovincialis in Ireland could have potential impacts on the ecology and the local ecosystem, and could also have socio-economical impacts because it could interfere with the aquaculture and food market of Mytilus edulis, which is a profitable industry in Ireland. As these three species are so morphologically similar, different molecular identification tools have been used for the identification of the Mytilus species complex. The established approach was defined in 1995 by Noah and colleagues, and it is based on a polymerase chain reaction, or PCR, which allows to produce a large number of copies of a DNA fragment of interest. They used a nuclear marker to target the adhesive foot protein gene to produce, with a gel electrophoresis visualization, size-diagnostic bands for each species. So I know here it is a bit small, but for Mytilus galloprovincialis, we have a band at 128 base pairs. For Mytilus edulis, we have one band at 180 base pairs. And instead, for the hybrids, you can see the presence of two bands. And this is how the species are identified. Now, more recently, SNP approaches have been tested. They use single nucleotide polymorphism markers, or SNP markers, and they are a genome-wide approach, so they target multiple genes. In 2018, Wilson and colleagues published a panel of 12 diagnostic SNP markers for the Mytilus species complex, and this image represents an outcome of a SNP genotyping approach. SNP genotyping allows to identify the base that is present at each polymorphic site. So in this case, we have the individuals in columns, and you can see that for each individual, there are 12 markers instead of just one band, like in the established approach. So therefore, the SNP approach has a higher diagnostic and a higher discriminatory power. Another important technique that is being developed recently are microfluidic systems. This image shows a machine called Biomark HD that allows to that is a microfluidic system that allows to miniaturize biological techniques, and it provides a high throughput approach. So the research objectives of this project were twofold. On one side, we had a technical one that aimed to optimize and transpose the SNP approach to a microfluidic system. So we tested the 12 SNPs published by Wilson and colleagues and we tried different experimental conditions to see which ones worked best. Instead, the application side aimed to assess the taxonomic resolution of the SNP approach for the Mytilus species complex. In total, I worked with 120 samples that were made available to me by my supervisor. These were collected in 2020 by these 6 sites that you can see on the map, and they had been previously identified with the established approach, so we already knew the species of each sample. Here also you can see the Mytilus edulis in black, Mytilus galloprovincialis in white, and the hybrids in gray. And again, in the Irish Sea, there were only Mytilus edulis, but Mytilus galloprovincialis were seen at all other sites, with hybrids only in Bantry, so on the southeastern coast. We decided to apply the SNP approach to the same 120 samples, and this is just a brief summary of our experimental workflow. So the first step was DNA extraction and quantification. We used 3 different extraction kits. The first one was Chilex 100 resin, which produces a low-quality extract. Instead, QIAGEN Dionisi blood and tissue kit and ESNA tissue kit produce higher quality extracts. All the extracts were quantified using a qubit fluorometer. The second step was a preamplification. Although a preamplification step is recommended by a manufacturer, we also decided to test a few samples without the preamplification step. However, preamplification is useful because it produces a high number of copies of the DNA of interest. And so we ran a preamplification and then proceeded with 2 dilution factors. So 1 to 100 dilution and a 10-fold dilution using molecular grade water. And at this point, we were able to start the genotyping process using Biomark HD, which I showed you before, with an endpoint PCR. We chose 2 different throughput approaches, so a Flex 6 IFC and the 9696 dynamic array IFC, on which I will go more in detail in the next slide. On the Flex 6 IFC, we ran 120 samples with 36 replicates. Instead, on the 9696, we ran 53 samples with 13 replicates. So integrated fluidic circuits, or IFCs, are plastic supports that contain a central chip, which is this one, where nanoliter accommodating chambers will contain, during the reaction, the assays and the samples, and so that is where the PCR takes place. The Flex 6 genotyping IFC is formed by 6 partitions, each one with 12 assay wells and 12 sample wells. These are then conducted to the central chip, where 144 reactions per partition can take place at the same time. The 6 partitions can be used simultaneously or in different moments, and therefore the Flex 6 size IFC is very versatile. Instead, the 9696 dynamic array IFC has 96 wells for the assays and 96 wells for the samples. So it is a high throughput approach because over 9,000 reactions take place in each run. Both these IFCs were used for SNP genotyping, but the Biomark HD system has many other approaches, and I'm sure that Eugenio, who will be presenting after me, will give you some insight on some other interesting applications. With regard to data analysis, we use the R software. We use the AdaGenet package for multivariate statistics on genotyping data. We use the ER package to calculate the kappa coefficient of concordance between replicates, and we use ggplot2 for graphical representation. The SNP genotyping analysis software was used to analyze the data obtained from Biomark HD. Structure software was used for population genetic inference, and MegaX software was used for analysis of the sequencing data. Now, diving into the results, this first slide represents the DNA quantification results. So on the left, we have the DNA concentration expressed in nanograms per microliter with the 3 different extraction kits. So this represents the initial DNA concentration of the extracts. Instead, on the right, we have the DNA copies in each reaction chamber, so the nanochambers where the PCR reaction takes place. And here, these are divided by different experimental conditions. So we have the Chilex extract with preamplification and with the 2 dilution factors. Here we have the ESNA without preamplification. Here the ESNA with preamplification and the 2 dilution factors. And finally, the QIAGEN preamplification with a 1 to 100 dilution factor. With regards to the starting concentrations, QIAGEN produced significantly higher yields of DNA concentration. Instead, for the DNA copies per reaction chamber, both ESNA and QIAGEN kits produced the highest number of DNA copies. But we also noticed that the Helix extraction method produced a sufficient number of DNA copies to have a good reaction. Therefore, it can be used as a more cost-effective approach because the kit is less expensive. However, for all the cases, we do recommend a preamplification approach, especially because the only extract that didn't have preamplification produced a very low number of DNA copies per reaction chamber. And we also would recommend a 10-fold dilution approach because it reduces the quantity and the cost of the reagents necessary. Here instead, we have the SNP approach outcome. So these graphs represent PCAs, or principal component analysis. PCA is a multivariate statistical technique that allows to reduce the many categorical variables into fewer principal components. And in this case, this is a representation of all the individual's genotypes. So this is the same PCA, but color-coded by amplification condition or by extraction method. So here we have by amplification condition, the non-amp and the preamp. And instead here we have the 3 different extraction kits. And as you can see, it was not possible to group the individuals based on these experimental conditions. These 2 graphs also represent the same PCA, but in this case, on the left, we have the color coding by IFC type, and on the right, the color coding by species. Now, the IFC type also did not produce any grouping, but it is interesting to notice that we calculated the kappa coefficient of concordance between the replicates that we ran on the Flex 6 IFC and on the 9696 IFC. While the Flex 6 had very poor concordance, the 9696 IFC had very high concordance. So we can recommend it because it is a more reliable IFC. We did expect to see grouping in the species PCA because we already knew that the samples contain different species, and therefore we were hoping to see the same division replicated with the genotyping data. Instead, this was not possible. As you can see here, there is no grouping. We also decided to compare our data with the data that was publicly available from the Wilson study, from which we obtained also the SNP markers. So here you can see that the species genotyped by Wilson, so here we have Mytilus galloprovincialis, Mytilus edulis, and Mytilus trossulus, and they form 3 separate clusters as expected. Instead, the data from the present study does not divide into the 2 species, and also it forms a separate cluster compared to the other 3 species obtained in the Wilson study. In conclusion, our technical investigation part was able to advance recommendations for the SNP approach using a microfluidic system. So we were able to recommend different techniques and different experimental conditions. However, further optimization is required to resolve the taxonomic identification of the Mytilus species complex. We do recommend continuing studying the SNP approach because it could become an effective identification and monitoring tool, and it could also give a higher insight on hybridization and introgression events. In particular, we recommend testing a larger SNP panel because this would increase the number of markers, therefore also increasing the discriminatory and diagnostic potential of the methodology. I would like to thank you for your attention, and I would also like to thank Luca and Sergio for their incredible help during my thesis project, the MFSC and the BIM staff, and the European Maritime Fisheries Fund for financial support. I would also like to thank my IMBRC family and wish good luck to everyone who will be presenting this week. Thank you.